Good morning, everyone. Okay, let's get started. Today we're going to continue. Good morning, everyone. Thank you. Yeah. Today we're going to continue the mutation in a large range of the genome. So, for example, either the entire chromosome or a large portion of a chromosome. So last time we talked about the trisomics. So in the trisomics for a specific chromosome, there are 3 copies like this, and so during the meiosis, how these 3 copies of the same chromosome, the homologous chromosome, are being paired during the meiosis. So there are 2 possible ways to do this. Either the 3 chromosomes homologous are gonna form this trivalent like this, or 2 of them are gonna form a bivalent like this, and leaving the other one segregate independently. So these are the 2 possible ways to do this during the meiosis. Okay, so such that for this trisomy individual, so suppose we have like 3 copies of the chromosome, right? So if one locus on this chromosome is carrying a gene, and for example, in this trisomy, there's 1 copy of the capital A and 2 copies of the lowercase a like this. This is the genotype for this trisomy, and we are interested in the types of gametes that this trisomy individual can produce. So based on what we have learned on the previous slides, so we can easily derive the 3 situations, right? So if the first, the big A and the if the big A go together with the first little a into 1 cell, then you're gonna get these 2 possible gametes, right? And if the big A will enter the same cell with the second copy of the little a, so then we get the same gametes like this, right? So if the 2 little a's come together into 1 gamete, then we're gonna get these 2 possible gametes. So if you summarize these 3 possibilities, then we can easily derive these results. So, okay, let me erase this first. Okay. So if you summarize these 3 possibilities, then you're gonna see, so there are 4 possible gametes that this trisomy individual can produce. Either the gametes with a capital A, right? This is 1 type, or little a, or big A and small a like this, or just 2 little a's, so like this. And the ratio of these 4 possible gametes is like this. So 1 out of 6, 2 out of 6, 2 out of 6, and 1 out of 6. So this is very important. Why this is important? Because we have a final, right? Final exam, and on the final exam, so this will be a topic to be covered. So I hope everyone can understand this, and I'm going to show you example. That example could be a problem on your final exam. Okay. So suppose we have a trisomic individual. So the genotype for the triploid. So the genotype for this trisomic or triploid is like this. All right. We have 3 copies of this gene, right? Big A, and small a. And the question could be like, what is the probability of this individual, the trisomic or triploid individual of genotype like this will produce progeny of genotype like this. So if we do the intercross, okay, between these genotypes. So first, we need to understand, so this individual, trisomic individual of this genotype is going to produce these four possible gametes here, right? And if you summarize this will give you the gamete frequency. So this is a little bit different from what I presented in the previous slide. So in that case, the genotype is A, little a, and a little a. So here we have a slightly different genotype, right? We have a big A, and a small a. So if you understand how this individual produced different types of gametes, so you can easily derive this gamete frequency here. So 2 out of 6 of this genotype gamete, and this 2 out of 6, 1 out of 6, and 1 out of 6. And then, so you need to apply a little bit statistical probability theorem to help you to get the right answer to calculate this probability. Okay, so all right, so we need to calculate the genotype of this probability, right? So how do we form a zygote with this genotype? So either we get a single little a from one parent, like this. Then we need another gamete with double capital A, right? So this is one possibility to generate a genotype like this. Okay, and there's another possibility is, okay, so we got the gamete of a single capital A from one parent, but we got another gamete with the big A and the small a gamete from the second parent. So there are two possible ways to do this. Then we're going to use the addition rule here. And so for each possible ways, we use the multiplication rule in statistics, right? So if you get this, and if you plug in the numbers here, so this will be the probability. So this will be the answer to this question. So that's the probability of this triploid or trisomic individual of this genotype will produce the same genotype if we do the cross between these two, the parents of this genotype, right? So any questions here? So this is very, very important. So first, so I could give you some different genotypes here, but based on the information I give you in the final exam, so you need to figure out how many types of gametes can this individual produce, trisomic individual produce, and what is the gamete frequency. So this is what you need to figure out in the final exam. And then if I ask you to calculate the probability for a progeny of a specific genotype, not necessarily to be this one, I could ask a different genotypes for you to calculate, and then you need to use this, something like this, the formula like this, to calculate the answer. All right. Yeah, I hope this is clear. Okay. So by the way, for the final exam, we used to have so for final exam, we're gonna use a different form for the questions. So because we need to get the grades back to you as soon as possible after the final exam. So we're gonna only use the multiple choices for the final exam. If I remember correctly, in the past we often gave students 25 questions, multiple choice questions. So yeah, we will do the same thing this time. But still, like, because we because we cannot have the exam in the classroom, so we will use Zoom or UJA system for the final exam, like the midterms, right? Okay, so last time we very briefly described how we can create the tetraploid from the diploid, right? So if this process is done within the same species, so then this polyploidy will be called the autopolyploid. So we're going to talk about this a little bit later, because there's another allopolyploid. So that's different definition for when compared to the auto, because allo means like we still form an individual with polyploidy, but the different sets of chromosomes may came from more than one species. So this is the difference between these two concepts here. So let's look at these slides to see how we can create a tetraploid from a diploid. So this, for example, this is a diploid. So for each chromosome, we have 2 copies, right? Okay. And so in the mitosis, this is not meiosis, it's in the mitosis. So every chromosome is gonna lining up along the cell equator, and the 2 homologous, two sister chromatids will be pulled away by the spindle fiber, right? And then so like this. So this is the normal way. So if the 2 sister chromatids are pulled away into 2 different daughter cells, then the ploidy will remain. Okay. However, if we use some specific chemical here, for example, the colchicine, if we apply this chemical to the cell, so the cell cannot go through the normal mitosis because this chemical can disrupt the formation of the spindle fibers. Then we see the DNA duplication. The chromosome, each chromosome has been duplicated into 2, but if there's no cell division, then you're going to see what? The consequence is like in the final resultant cell, you're going to see the ploidy being doubled. So by doing this, we can create a tetraploid from a diploid. So this is how we can do this. This is one way. Of course, there are some other ways that people can use to create polyploidy individuals. All right, so this is the way how we do this. So then how do we create a triploid? So that's easy, right? Because we just learned how we can create a tetraploid from diploid using the colchicine. So if we successfully create a tetraploid, then we can easily cross the tetraploid with the diploid to produce a triploid here. The same thing here. So this is how we, okay. Actually, so no. So is this concept similar to the heterokaryons where the cells have multiple genetic nuclei? This is a little bit different because so when we talk about good question though. So let me go back to the erase this first. Okay. So oops. So when we talk about the heterokaryon, so we mean, so usually we mean like, so this is one neurospora with a nucleus, right? This is another neurospora with another nucleus. All right, so this nucleus may have a set of like genetic material. This one may have a different set of genetic material, right? Different genotypes, for example. So the heterokaryon means these 2 cells will form together will form a cell, will fuse together to form a cell which has both nuclei. And so, for example, if so we have a question. We had a question in the midterm, right? So, for example, if this Neurospora strain is a mutant, so for example, there's a pathway like from the precursor. Okay, so it can produce A, and it can produce B, and finally, this can produce something useful for the growth of the Neurospora. Okay, and so there's an enzyme here, there's an enzyme here, and an enzyme here. So enzyme 1, let's say, enzyme 2, and enzyme 3. Okay. All right, so for this mutant, for example, if this enzyme has something wrong because of the mutant, so that means like this strain cannot grow on the minimum medium by itself, right? Because of the deficiency of this enzyme. If, for example, this strain has a different problem. So, for example, if this enzyme 2 has been mutated, so this strain cannot by itself grow on the minimum medium as well. However, if we can fuse these two into this hetero carrion like this, so this cell containing like more than one nuclei, right? And what happens here? So now this fused thing can grow by itself on the minimum medium. Why? Because although this one cannot produce functional enzyme 1, but this one can, right? This one can produce the enzyme 1 functional. However, this one cannot produce the functional enzyme 2, but this one can. So this one can. So if you put them together, if you fuse them together to form a heterocarrion, so the entire pathway becomes a functional. So that's why this thing can grow by itself on the minimum medium. However, this is a different situation because so this came from the same nucleus, right? So there's no like different nucleus. So this is a different concept here. So we just starting from one nucleus and try to use this chemical colchicine to disrupt the formation of the spindle fiber. Then the resultant cell still has a single nucleus. So this is different from this concept. All right, very good. Thank you for asking this. I hope this is clear. Okay, thank you. All right, so let's look at an example here. Okay, so we have learned, so we can use the colchicine to produce a 4N, the tetraploid from the 2N, right? Once we have the 4N, so we can easily cross 4N with the 2N to generate a 3N. So the 4N individual can produce a 2N gamete, and the 2N individual can produce a 1N gamete, and this two can produce a 3N seeds, right? The watermelon. Now, if we grow this 3N seeds into the plants, the watermelon, and we cross this 3N watermelon, so if we intercross this triploid, the 3N watermelon, so let's see what happened here. So you're gonna see the watermelon without seeds here, the seedless. So why is that? Why is that? All right, let's look at it. So we have a first parent is 3N, and the second parent is 3N. So if the cross between these two 3N parent can produce seeds. So this is one possibility. So one possibility is like this individual, this parent will produce a 1N gamete, and also this parent will produce 1N gamete. So only if this is the case, then you're going to see the 2N seeds, right? Otherwise, so you cannot see the seeds. So why is that? Let's look at this slide. Okay, so suppose this is a 3N parent, and for each chromosome, I only show you 4 chromosomes here. Actually, in the watermelon, there are 11 chromosomes, but because of the space, so I just use a 4 as an example here. So this is a 3N parent. Okay, now so this parent go through the meiosis to produce gametes. So it's very likely to produce a gametes like this, right? So because each chromosome is going to segregate independently. So we have learned this before based on Mendelian's law, right? All right, so for the first chromosome, probably this gamete only has 1 copy. But for this chromosome, this gamete may have 2 copies. All right, and so on. Definitely, this is not a 1N gamete, right? And also, it could be end up with this gamete. So this one has 2 copies, but this one has 1 copy. And so if you sample a lot of gametes from this 3N parents, majority of the gametes will look like this. So they are not 1N. So that's why you do not. Usually, we do not see the seeds in the watermelon. So only if this parent can produce a 1N gametes like this. So for each chromosome, there's only 1 copy. Then it's possible to form a watermelon with seeds, right? So let's see. So let's calculate what's the probability that any 3N parents can produce a 1N gamete. So that's the probability. So it's extremely low, right? All right. So one 3N parent will produce 1N gametes with this probability, and we need the same thing for the other parents as well. So these parents can should also produce a 1N gametes. So that's why so we would like to multiply these numbers, or by 2. But we will calculate these numbers to the power of 2 here. So that's a extremely small thing, very tiny little chance, right? So that's why you understand. So if we cross a 3N watermelon with another 3N watermelon, so we usually get this, so the watermelon without seeds. So this is a good feature for customers. All right. So I hope this is clear. Okay. And okay, so now we need to learn. So for the tetraploid, for each chromosome, we have 4 copies, right? So during the meiosis, how these 4 copies of the chromosome will be paired together. So there are 3 possible ways. Either 2 of them form a bivalence, and there are 2 bivalents here. So each one is going to segregate independently, and or 4 can form a single, like a quadrivalent, right? So this is not a possible way. So only these 2 are possible, and either way can produce gametes of 2N. Okay, from this tetraploid. Okay, so this is example, but this is a little bit tricky because I promise I will not put this on the test, but it's good for you for you to practice this one. But for the a triploid. So this is for the tetraploid, right? For the triploid, definitely that's required. So it's very likely for me to put a question about the triploid individual in the final exam, but tetraploid, so this is too complicated. So I'm gonna just show you how to do this. So for example, if we have a parents like this and a parents like this, so the tetraploid parent will produce a 2N gametes, right? And this can form the F1 individual, so the genotypes. So what if we intercross the F1 progeny of this genotypes? So it's a little bit complicated. First, we need to figure out the same strategy. So first, we need to figure out how many types of gametes can be produced from this from the parents of this genotype. So this is the way you think about this. And okay, so for each parent, 3 possible gametes can be produced. 3 possible 2N gametes can be produced from this F1 parent, and the frequency associated, the gamete frequency associated with these 3 types of gametes is like this. So 1/6, 2/3, and 1/6. So this is for one parent. So right now we are expert of using Punnett square, right? This is something very similar to the Punnett square. You use 1 dimension to represent the possible gametes produced from one parent, and you're going to use the other dimension to represent the possible gametes produced by the second parent, and those are the possible progeny genotype, the genotypes for the progeny produced from this cross, right? And then if you summarize this table, so you can easily get the probability and to help you to get the to do the calculation. All right. So again, so this is a little bit too advanced. So we will not see the problem like this on the test. But for the triploid, let me go back to here. So for this, so definitely this is important. So everybody should understand the biology behind this. And if I give you a question like this, so you should be able to work to find out the answer to this problem. Okay. Okay. All right. So now 2 important concepts here. So we have talked about the autopolyploids, right? If all the chromosome sets come from the same species, so then that polyploid will be the autopolyploid. However, sometimes if for an individual polyploid, if this individual has 2 or more chromosome sets coming from 2 or more species, so in this case, this polyploid will be called as a allopolyploid. So this is example here. So if you cross, so if a donkey is mated by, so if you cross a donkey with a donkey, so then of course the progeny will be a donkey, right? So that's the autopolyploid, because for this individual, for this donkey, the 2 sets of chromosomes came from the same species, donkey and donkey. So the same for horse. So if you made a horse with another horse, so you can get a autopolyploid horse. However, if a donkey mates a horse, or if then you're gonna get a individual. For this individual, 2 sets of chromosomes came from 2 different species, and in this case, this polyploid is going to call the allopolyploid, right? So autopolyploid means within the cross within a species, one species. But allopolyploid means like the progeny is produced by the cross between two species or more. Okay. Okay. So so in a horse, there are so diploid and horse, there are a total of 64 chromosomes, which means like there are 32 different chromosomes, but it's 2n, so that's why the total number is 64. For donkey, it's a little bit different. It's 62. All right. However, so if we cross, let's say mule is here. So if we cross a male donkey with a female horse, we're gonna get the mule, right? The mule is a allopolyploid. So the total number of the chromosome in a mule is 63. So the same for the hinny. Okay. And so what's the difference? So as you can see, so in a mule, okay, so there are 63 chromosomes in a mule, and 32 chromosomes came from horse mom, and 31 chromosomes came from the donkey dad. So they have a slightly, so if you compare the mule and the hinny with the regular horse or donkey, so you're gonna see slightly different phenotypes here, right? The major thing is like they are stereo, right? They cannot produce, usually they cannot produce progeny because of this specific genetic setting, right? However, all right, so this is the genetic setting for this for a different situation, right? But once in a blue moon, so the blue moon means there's very very small chance, tiny chance, a female mule and a male horse can produce a offspring. So why is that? Because so now let's see these two parents, a female mule cross a male horse. Okay, so male horse is 64, right? Female mule should be 32 horse chromosomes plus 31 donkey chromosomes, right? All right. So what's the situation that this cross can produce offspring? So let's look at the horse. The horse is regular, right? So it can always produce 32 chromosomes, horse chromosomes. However, so the gamete product from this female mule will be very quite diverse. So you can expect to see, we expect to see all kinds of possible chromosomes in gametes, right? So a lot of chromosomes, a lot of possible different possible gametes. For example, for this one, so it can have like maybe let's see gametes like 16 horse chromosome and 15 donkey chromosome, something like this. So this definitely cannot come together with the regular horse gametes to form a progeny, right? No, and you could see like maybe 8 horse chromosomes and let's see, maybe 24 donkey chromosomes, something. No. Yes. Okay. Maybe, yeah. So this is another possibility. So you can see lots of gametes like this. So they cannot form gametes with the regular host gametes, right? Only if this female mule produce a gametes where the 32 host chromosome are represented in this gametes. And okay, then if this female mule produce a gamete like this, so these two can come together to form a horse progeny, right? But as you can see, the chance of this is very, very small, because the chance of getting the irregular gametes will be extremely large, but the chance for this female mule to produce a gametes with all 32 host chromosomes will be very tiny. The chance is very tiny. So that's why once in a blue moon, so a female mule and a male horse can produce an offspring. So, oops. And so this is the chance. So probability of that one gamete with 32 host chromosomes will be like this. So this is 1 out of 10 billion. So this chance is really, really like small. And all right, so this is the case where this will happen. Okay, so blue moon. Okay, this is my favorite brand for the beer. All right. Okay. All right. So let me see. So okay. So we have time, so we can continue to talk about. So let me give you a brief summary here. So at the first part of this lecture, we talked about the specific inheritance pattern for the triploid. So this is important. We talk about the inheritance pattern for the tetraploid, but that's a little bit too advanced. So we're not gonna worry about that in the final exam. But again, the triploid or trisomic should be very, very important, and everybody should understand and know the biology behind this one. And definitely, you're gonna see some questions like this in the final. Okay, so yeah, get yourself well prepared for this, for the problem like this type. Okay, and okay, so let's continue. So now we're going to talk about change in a large chromosomal segment. So not the entire chromosome or chromosome sets, just a large segment within a chromosome, right? So this slide just summarize the regular, very standard situation for the large-scale changes in the chromosome structure. So in the middle, this is the wild type sequence. So this is a chromosome. We use like different color to represent the different portion of this chromosome. So this is the wild type, means like this is the standard genetic setup for this chromosome. So if you compare this one with this one, what do you see? What the difference did you see? So yes, we see something is missing, right? The brown region is missing. So definitely, so in this situation, so we call this is a deletion, because we see some DNA segment, a large chromosomal segment has been deleted from the wild type. So that's the deletion. All right, so if you compare this one with this one, so you see something has been deleted, but you see something extra, right? It's a different. So actually, this segment of the chromosome is translocated from other chromosomes. So because of the exchange, DNA exchange between this chromosome and some other chromosome, then if this is the case, we say this is the translocation, right? Okay, and so if you compare this one with the wild type, you see the order of the brown one and the green one has been changed, right? So that means like this region has been inversed. So this is the inversion. And so if you compare this one with this one, so this one is significantly longer than the wild type one, right? Right? Because of what? Because this region, the brown region, has been duplicated. You see like 2 copies of this brown region in this situation. So that's the duplication. Okay, so if in a cell you see 2 copies of this wild type, right? That's the extra chromosome, right? The trisomic. So, and if you in a cell you see like 1 chromosome is missing, so that's the monosomic, right? So we have covered this before, and so the entire chromosome missing or entire chromosome extra, so that's probably due to the non-disjunction non-disjunction during the meiosis, right? The gamete formation. All right. So some student asked me before the midterm 2, so whether the midterm 2 will be cumulative. Yes. So our test will be cumulative. So because our class is taught in a progressive way. So at the beginning, the first part before the midterm 1, we learn very basic genetic inheritance rules, and later on, so we migrate into some complicated situation, so which are built upon the previous part. So if you want to understand well for the later part, so definitely you need the basic knowledge from the first part, right? So the same for the final. So the final will be cumulative as well. So we're going to cover all the stuff we have learned before, but with an emphasis on the later part, the third part. But still, like, don't be surprised if you see some like old questions on the final, because that's definitely possible and necessary. Because all the stuff are associated with each other, and what you have learned in the third part will be built upon the knowledge you acquired from the first 2 parts, right? Okay. All right. So so this slide just shows the mechanism that cause different different changes in the chromosomal the chromosomal structure change in the previous slide. So deletion. So this is one way. Deletion. So this is a single chromosome. If like there's a like breakage and rejoin. So if there's a breakage here, and these 2 chromosomes if rejoined together, then you're gonna see right. So this region has been deleted. If there are 4 genes on this on the initial or original chromosome, then after the deletion, you only see 2 of them. So the other 2 has been deleted. So this is one way that can form the deletion. And another way to form a deletion is through the crossing over between the repetitive DNA. So suppose this is the original chromosome, and so if there's a DNA sequence here, and there's another identical DNA sequence here. So so if this is the case, these 2 similar region or identical region can be paired together like this, and the crossover could occur between this within this chromosome. And then what is the consequence of the pairing of this repetitive DNA and the crossing over? So the results is like, okay, so it will form a loop like this, and this small DNA will be lost and leaving the resultant chromosome like this. So, and as you can see, so after the crossing over and the pairing and the crossing over, the DNA, the chromosome segment containing gene 2 and 3 is gone, so it has been lost, and you only have like gene 1 and 4 on the chromosome. So this is the mechanism to produce the deletion, and the deletion and the duplication. So this could be like this. So all right, so we have like so the first one is within the same chromosome, just single chromosome, right? But the second one is between two chromosomes. So if the two homologous chromosomes are pairing together, and if the crossover between these two homologous chromosomes are not even, so if, for example, so if the crossover between these two chromosomes occurs between here and here, then what happened? So after the crossover, you're gonna see for one chromosome, gene 3 has been deleted, right? But for the other chromosome, after the crossing over, you're gonna see there is an extra copy of the gene 3. So this is the one way that that can produce deletion, either both deletion and the duplication during the between the two homologous chromosomes. So the same thing here, right? All right. So sorry. Actually, let me see. Okay. So yes. So this is the same. So if there's like a repetitive DNA, so this is another mechanism. So for example, this is one homologous chromosome, the other homologous chromosome. However, so there are two regions here. They are identical, so that's the repetitive DNA. So if the crossing over between these two repetitive DNA, then what's the results? You're gonna see for this one, after the crossing over, the copy of the 2 and 3 will be deleted. But for this one, you're gonna see two copies of the gene 2 and 3, right? So this is easy to understand the mechanism. And for the inversion, yeah, it's easy. So if there's a breakage here, and if the rejoining is occurring like this, so if this end is rejoining with this end, and this end rejoining with this end, then the result is like you're gonna see this region in the middle carrying gene 2 and 3 has been inversed. So this could be due to the repetitive DNA as well. If there are two regions represent the repetitive DNA, but these two, the orientation of these two repetitive DNA are different, then it can be paired in this way. And after the crossing over, you're going to see the region in the middle has been reversed, has been inversed, right? And the translocation means the DNA exchange between non-homologous chromosomes. So compared to this one is the DNA exchange between two homologous chromosomes, right? However, the translocation means the DNA exchange between non-homologous chromosomes. So we use different color to represent here. So this is, for example, maybe the chromosome 1. It has totally different genes. It has gene 1, 2, 3, 4. But this might be the chromosome number 8. So it has a gene 5, 6, 7, 8, and 9, 10. So if there's a DNA exchange between these two non-homologous chromosomes, you're going to see this. The translocation, right? This could also be due to the crossing over between the repetitive DNA. So the same mechanism here. All right. Okay. So so next we're gonna oops. Let's see. Can you explain deletion and the duplication? Okay, sure. deletion and the duplication. So this one, right? Okay. All right. So this one, let's look at this two. So the first one is the simplest way, is it only involves a single chromosome, right? A single chromosome. So the chromosome just break itself, and if it this end is joining with this end, then the region in the middle will be lost. So that's the deletion, simple one. The same here. So we are talking about just one chromosome, single chromosome. But within the single chromosome, you have two regions. The DNA sequence for these two regions are identical. So we say this is the repetitive DNA. And these two identical repetitive DNA can be paired like this. And so just like the pairing, right? The regular chromosome pairing, because they are identical. And if there's a crossover between these two, so this will result in this. So there's a loop containing gene 2 and 3. So it is gone. And the resultant chromosome will be like this. So you're going to see some part missing from this mechanism. So this is within a single chromosome. But the second mechanism, deletion and the duplication, this happens between two homologous chromosomes. So we have chromosome 1, homologous chromosome. They are the same chromosome, right? They should be paired together and doing the crossover. But for most of the situation, the crossover between these two homologous chromosomes should be even. What is even? So because the crossover should occur at the same locus. But however, sometimes, yeah, if this is not true, so for example, if the crossover occurring at a different location, so the breakage on the first one is between 2 and 3, right? But the breakage for the second homologous chromosome is between 3 and 4. So if the crossover occurs like this, then what you're going to see, so this the resultant chromosome will be, one is this, right? This one, and the other one is this one. So the first one like this. So if you go this way, so this will become like 1, 2, 4. So 3 is missing. But for this one, the longer one, you're gonna start from here and here. Oops. So you're gonna see gene 3 twice, right? Because of this uneven crossover. So for this uneven crossover, you're gonna see the other chromosome carrying 2 copies of the gene 3. So that's the duplication, and that's the deletion. And if this is the mechanism, you're gonna see this crossing over will produce deletion and the duplication simultaneously. So you're gonna see the deletion and the duplication together. This is the same, right? So we see the crossover occurs between 2 homologous chromosomes. For the first homologous chromosome, there are 2 repetitive region, right? For the second one, the same. In the common situation, so this one is going to pairing with this one. This one is going to be pairing with this one. But sometimes, like if this region pairs with this one, so if the crossover occurring between these 2 regions, then the problem comes, right? Okay, very good. Okay. So this is important because in the final, I may give you a question like this to ask you to multiple choice so I may ask you some resultant gametes with a specific genotypes, and then I will ask you what's the mechanism behind this, right? Okay, so I think, yeah, this will be the, so I'm gonna briefly introduce this for you. So we're gonna talk about this in detail next time. Okay, so because deletion is very important. So next we're gonna talk about deletion. So, all right, let me see. So suppose this is the wild type. So this the wild type chromosome. You're gonna see the intact genes on this chromosome, right? A, B, C, D, E, F, G, H. All right. If there's a deletion here, so the deletion including one end of the chromosome, then this deletion is called the terminal deletion. So you're gonna see it. One end is missing. Okay. So if the deletion is in the middle, and the chromosome is going to be rejoined like this, so then this is called the interstitial deletion. So there are two possible ways to form the deletion in the chromosome, right? So depending on the location. Okay, and so this is an important experiment. So we're going to talk about this in details next time. So this is a way that we can use the deletion to help us to do the mapping to figure out the order of different genes on the same chromosome. Suppose we have like 7 genes on the chromosome, A, B, C, D, E, F, G, like this. So we can form a heterozygous, right, for each locus. So for each locus, we have a heterozygous. All right, and for each locus, if the two alleles are complete dominance. So for example, this is a wild type, this is a mutant. So then the entire individual should be the wild type, right? Because the wild type is the dominant. So all wild type is dominant to the mutant. Okay, now suppose we introduce some deletion in this chromosome. So as you can see, this region has been deletion, right? And okay, so then what will be the phenotype after the deletion? It's obvious, right? So within the region without deletions, of course, the phenotype will be wild type because there exist the wild type alleles, right? There's no problem. So each gene is controlling one phenotype. So without the deletion, there exists the wild type alleles for that gene. So then we're going to see wild type for those phenotypes, right? Plus. However, because of the deletion, so 2 wild type alleles for B and C genes are missing. Then this is the pseudo dominance. So we have talked about this before, right? Hemizygous, because of the deletion, these 2 genes only have 1 copies, and because for these 2 genes, only the mutant allele remains. Then for this 2 genes, you're going to see the mutant phenotype because of the deletion. Does that make sense? So this is the mechanism. This is the mechanism we're going to use for the next time. So this is a very good tool we can introduce. So if we know the genes are on the same chromosome, but if we want to figure out the order of the genes, so this is one way. Another option that we can use deletion mutant to help us to figure out the order of the genes. So for example, so this is a fruit fly Drosophila example. So this is a specific chromosome, and on this region there are a lot of genes here. So if you see the dark band, that probably represents different genes. And we introduced 13 different lines, so different mutant lines. Each line involves a specific deletion. So for example, the first line named this 258-11. So this mutant line has a deletion in this region. So we can use this to help us to derive the map of the genes on this chromosome, right? So something like this. So next time we're going to talk about this. So in the final, this could be it's very likely, a question like this will appear on the final exam. And I'm going to give you the question like this. So this is a chromosome, and we knew like 6 loci containing 6 genes here, but we do, and these 6 genes are A, B, C, D, E, F. But so at the beginning we do not know the order of this. Okay, we do not know the order, so we need to figure out where A stands. So the data will be given like this. So we have the deletion 1. So this is the first mutant line due to the deletion 2, deletion 3, deletion 4, and deletion 5. And based on the deletion region and the phenotypes. So for example, for deletion 1, so we see wild type for gene D, E, F, but mutant for A, B, C. Then immediately we knew, okay, so these 3 regions should be A, B, C, and this should be D, E, F, right? But which one is A, which one is B, we don't know. So we need more information to help us, right? So let's look at the deletion 2. So deletion 2 actually covers this loci and this locus and this locus. All right. So these two loci are in common with the deletion 1, but the difference is this region, right? So if we compare deletion 1 and deletion 2, what's the difference? The difference is here. So these two are identical. The overlapping region, which is the B 2 and 3, right? And only this is different. So immediately, based on this information, we can know, we can conclude the first locus should be gene B, right? So it's easy. All right, so I'm gonna stop here because we're gonna talk about this more next time, and this is a very important point. Definitely, I'm gonna put some question like this in the final exam. All right, I'm gonna stop here. So if you have any questions so far, we can discuss. We can use this time, the time to discuss your question. Right.
I had a question, Professor.
Mm-hmm.
The final, I just wanted to double check. It's cumulative, right?
Yes.
Okay, thank you.
Sure. Yes. But with the emphasis on the later part, we have 3 parts, right? Part 1 is mainly for the midterm 1. Part 2 is mainly for the midterm 2. But also, we're gonna use the knowledge back to the part 1. And the final, the same. Is right now we're learning new stuff, but this is the knowledge of the new stuff completely depend on the knowledge, the old knowledge we acquired from the first 2 parts, right? So yeah, that's right. So you don't be surprised to see some old questions on the final exam. So that's why. So it would be helpful if you have time. Please review the previous midterms and the quiz. That can help.
Thank you.
Sure. Any other questions? All right. So this is important again. We use the deletion to help us to do the mapping. So so in the past we have learned how we can use the test cross, right? 3-point test cross to help us to figure out the order of 3 genes on the same chromosome. So here, so similar things, but we use different mechanism. We use the deletion mutant to help us to figure out the order of the genes on the same chromosome. So we're going to talk about this next time. All right. So if you do not have questions, we're going to stop here and see you next week. Tomorrow, I will have office hours. In case you have questions, bring your questions to my office hour. Right? Thank you, everyone.